So it's really important you guys ask questions, right? Questions are really critical. As you say, when we introduce ourselves, you'll see just how broad and diverse the class is. We have an extraordinary range of backgrounds. So there'll be some ideas or topics that some people haven't been exposed to. And it's critically important, if I talk about a concept you haven't seen before, please ask me to go into it in detail, right? Do not be bashful. We have a very diverse group. So for example, it may be something on eigenvalue if you're not a mathematician. It may be something on biology if you're a mathematician. So that's an important point to stress is this course is extremely broad and covers a lot of stuff. So it's really important that if you have questions, you ask right up front. Okay? So that's the one I want to stress it right up front. Number 2 is, have people found where the lectures for this course are? So if you go to the Nimbus page, you're supposed to have this set up here. Maybe I'll go back. Well, okay. Anyhow, on the Nimbus page, there's a mysterious link called World Press Spatial Statistics. They didn't change the link. That's actually us. It's now changed. How do we go back to that page, by the way? Didn't we have it up here? I don't see any. Okay. And so if you click on that, there are PDFs for all the lectures in this course I've been putting up one by one. That's also a forum. So basically here you are, you log into here. Oh, that's the streaming, never mind. Yeah, we'll ignore that. And that's also a forum for you to have general posts, questions amongst yourself, et cetera. So here's the web page. And if you just scroll down here, scroll, there it is. See, now it's the search for selection WordPress site. So you click on that, and it comes up with this. And if you scroll down a little bit more, recent posts, and actually all the lectures are posted on here. They're just, more recent than that. So if you open them up, they're PDFs, for example, for all the lectures, and you can just click on those. You're more than welcome to load them on your laptop and bring them to class or not. One way to think about a course like this is it's a fire hose of information. We're throwing so much stuff at you that what you want to come away with is basic concepts and also where to find some of this material. And the lecture notes are a good source to find some of this material, as we'll talk about. Okay? So let me kind of set the stage, tell you something about myself, and have you go around and introduce yourselves in a little bit of details we'll talk about in a second. So the focus of this course is selection. All biologists are fascinated with selection. We look at natural diversity, and we look at some unusual feature, and we concoct all these scenarios for how selection has shaped that, what the forces are. That's kind of Bravo Sierra BS. We need to have a formal framework for actually asking, is a certain trait or character or feature under selection? How do we tell? And there's a huge body of literature that's scattered around that talks about this. And basically it comes in 4 different flavors that we'll talk about. Flavor number 1, I'm looking in the fossil record. I've got a time series. Or I have genomic data. I'm looking at levels of gene expression divergence between 2 sibling species. So I have a pattern of phenotypic variation. And a standard test to ask, is that pattern of variation show too much directional selection or too little stabilizing selection relative to the pattern I expect under drift? There's a lot of classes that go in there, and these are nice. We'll talk about these today. These unite, for example, fossil data, temporal or spatial data on mean phenotypes, and genomic data. So these 3 very different fields come together. Number 2, this is relatively recent. I might have markers for specific traits. Markers I obtain by a QTL or a GWAS analysis. These are methods to try to basically tag chromosome regions that are involved in variation in a trait. And I ask, given those markers, which are given by specific traits, do I see something? For example, is there an excess of, let's say, plus alleles? We'll also talk about this category of tests today. Then we have a huge category of tests based upon marker data alone. So these 2, this is looking at a trait, this is using a trait to get markers. These are simply looking at markers. And so most of the literature you read on looking for signatures of selection are these genomic-based tests that, for example, use polymorphism data or divergence data, and examples are Tajimas-D, singleton density score test, McDonald-Kreitman HKA test. There's like 120 tests in here. There's a lot of tests in here. So trait-based, using the traits to get markers, entirely marker, and then there's the ecological data, which is if I go in the field and I actually measure how many mates does this phenotype have, how does this phenotype survive, I can actually then, with estimates of individual fitness, get direct associations between trait value and fitness. These things are all operating over different scales. This is ecology, this is molecular population genetics, this is paleontology, genomics, this is somewhere in between. So all of these different concepts are how we can detect selection. And when I proposed this course, we had thought that our book would be out in time. It's being printed literally on Friday. So what I'm going to do basically is I'll be giving you an overview. And the way to think about it is I'm giving you a big giant box of tools, right? So what you want to do as a biologist or a modeler is when you're faced with a problem you want to solve that problem by using the problem to tell you which tools to use. You don't want to use the tools to tell you which problem to attack. And the larger your toolbox is, the more flexibility you'll have in creative ways to address problems. And my job is to show you what that toolbox is, sort of hold up all the tools. And if you want to use them, you actually then, since you know what a tool is, you can then go back and look up in detail, and the details basically sort of found in here. And the course is only covering a small part of this, chapters 8 through 10, 12, and 29 through 30. So that's kind of the overview, where we're headed. Okay? Any questions or comments so far? And again, don't be bashful. That's kind of where we're headed. Yes?
So it's being live streamed. Will we be able to go back and watch a video of the lecture? Yes. Yes.
Sure. So you can see how bright and shiny my bald spot is when the lights get on. That's really exciting. I always cringe when I see that. So I just, but I'm not going to wear a hat. I should wear like a volunteer hat or something like that. So let me give you some just kind of background about myself before I have you introduce yourselves. So I have an unusual path, academic path. My actual undergraduate major is in mathematical population biology under Michael Trelli at Davis. Davis had an individual majors program, and I figured out very early on, hey, I can take all the fun courses I want, like phylogenetics and partial differential equations, and not have to worry about so many of those other requirements by making an individual major. So I did that. Then I got my PhD in University of Washington under Jill Felsenstein, who did a lot of stuff in phylogeny, although I took him for population genetics. I trained at University of Chicago under Tom Nagalaki and Russ Landy. Tom is not as well known. He's a very hardcore theoretical population geneticist. Russ Landy is one of the founders of kind of the Chicago School of modern evolutionary quantitative genetics. And I've been in Arizona for 30 years. And to give you some idea about kind of the areas I span, my main appointment is in ecology and evolutionary biology, but I've also got appointments in public health, plant science, animal science, and molecular cellular biology. Because I do a bunch of weird things, right? What are my interests? Evolutionary biology. I am a biologist interested in diversity of life and how it got that way. That's my basic interest. So genetics of complex traits is an area that I'm very interested in. Statistic and mathematical modeling. Plant and animal breeding are really interesting examples of applied evolution with enormous data sets. I mean, the best data sets on the planet are in plant and animal breeding in terms of looking at long-term evolution. I do a lot of teaching. I've taught in 25 countries. I added Uruguay this year. That was fun. And I teach a bunch of topics all related to this idea of evolution on genetics of complex traits. And a key idea in evolution of genetics on complex traits is how do those traits change over time? What are the forces behind them changing? I always like to throw a fun fact in. So I have an odd hobby. I do moth collecting. So this is the Chiricahua Mountains in Arizona at about 8,000 feet. This funny looking thing has 2 mercury vapor lights. You run it off a generator, and when our summer monsoons hit, it looks like that. And so when I look at biology, a long, long time ago, a colleague of mine, unfortunately I forget who it was, said you should always have a group that you're really familiar with. So whenever you have a question in biology, you can go back and reframe it in a group that you're familiar with. And my group is moths in this case. And so if you look a bit closer, so these are species which are actually found in the east, and they're big and showy. But the interesting thing is this slide here. And if you're a biologist and look at that and go, what forces shape those patterns? But that's actually, you get drawn to the wrong thing, right? You get drawn to this beautiful Rothschilda cincta, this silk moth that's only found in southeastern Arizona. A different species comes in southern Texas. What you're missing is those right there. Those are 3 different species in the genus Matagamma. You can't tell them apart. You've got to basically dissect their genitalia to tell them apart. These little things down here, those are 9 species of different flower moths, the acontiines. What are all the forces generating those? So when I look at biology, I always frame it in the thing, when I'm out there in the field looking at stuff, I get really interesting things. That kind of gives you some background on me.